Welcome all in my lecture ITASSER server. Dear learner, 3D JIGSAW server is not available now. Hence, in place of that, we will learn the ITASSER server for protein structure modeling. ITASSER server is a widely used bioinformatics tool for predicting protein structure and function. ITASSER stands for Iterative Threading Assembly Refinement, and it combines several techniques to generate structural models and functional annotations for proteins. Here's an overview of its capabilities and how it works. ITASSER is primarily known for its protein structure prediction capabilities. It generates 3D structural models for a given protein sequences. This process involves several steps. I am going to explain one by one. First one is threading. ITASSER identifies structural templates from the protein data bank and aligns the target protein sequence to these templates using threading techniques. Second is model assembly. It assembles the initial structural models by borrowing information from the identified templates. Third one is model refinement. The server then refines the models through iterative simulations, optimizing the energy and overall quality of the structures. Next is model selection. After refinement, ITASSER ranks the models based on their quality, and the best model are presented to the user. Next one is function prediction. Beyond structural information, ITASSER can also provide functional insight into the protein. It does this by annotating the predicting structures and comparing them to know proteins with similar structures and functions. Next is ligand binding sites. ITASSER can predict potential ligand binding sites on the protein's surface, which is crucial for understanding its functional roles and potential interaction with other molecules. Next is protein-protein interaction sites. The server can also predict protein-protein interaction sites, helping researchers understand how the protein may interact with other biomolecules. Next is gene ontology annotation. ITASSER can assign gene ontology terms to the target protein, providing information about its cellular location, molecular function and biological processes it might be involved in. Next is visualization. The server offers visualization tools to explore the predicted 3D structures and associated functional annotations. To use the ITASSER server, you typically provide the protein sequence of interest and the server performs the prediction and analysis. You can access the ITASSER server through its web interface, which is user friendly and widely used by researchers in the field of structural biology and bioinformatics. Please note that the specifics of the ITASSER server, including its features, algorithms and web interface, may have evolved or changed since my last knowledge update in September 2021. Therefore, I recommend visiting the website or referring to the latest documentation for the most up-to-date information on how to use the server and its capabilities. Now, my colleague Jyotsna will explain this server in detail.
Hello everyone. In this lecture, we are going to discuss ITASSER web server. ITASSER stands for Iterative Threading Assembly Refinement. It is a unified platform for automated protein structure prediction. ITASSER server is an online platform that implements the ITASSER based algorithm for protein structure and function prediction. It allows academic users to automatically generate high quality model prediction of 3D structure and biological function of protein molecule from their amino acid sequence. Homology modeling, also known as comparative modeling, is a computational method used to predict the three-dimensional structure of protein based on the known structure of related protein. This approach relies on the assumption that protein with similar amino acid sequence share similar structure and function. Here are the key steps involved in homology modeling of a protein. Sequence retrieval. Obtain the amino acid sequence of the target protein for which you want to predict the 3D structure. This sequence is typically obtained through experimental techniques or from databases such as UniProt, Protein Data Bank, etc. Homolog identification. Search protein database, that is Protein Data Bank PDB, and sequence database, that is NCBI BLAST, to identify protein with known 3D structure that are similar to your target protein. The quality of the template is crucial for successful modeling. Alignment. Align the amino acid sequence of your query protein with the sequence of identified template protein. The alignment should consider not only sequence identity but also similarities in secondary structure and functional domains. Template selection. Choose one or more template structures that are most closely related to your target protein. The selection should be based on factors such as sequence identity, structural quality, and relevance to the target protein's function. Model building. Generate an initial 3D model of your query protein by transferring the coordinates of a corresponding atoms from the template structure to your target sequence. The model is built using the alignment information. Loop modeling. If there are a region in the target protein that are not covered by the template or have significant sequence variation, that is loops, these regions may need to be modeled separately using method like loop modeling or ab initio modeling. Model refinement. Refine the initial model to improve its quality. This can involve energy minimization, optimization of bond angles and dihedral angles, and addressing steric clashes. Validation. Assess the quality of final model using various validation tools and matrices such as Ramachandran plot, mole probability, and ProCheck. These checks help ensure that model conforms to acceptable structural standard. Visualization and analysis. Use molecular visualization software, that is PyMol, UCSF, Chimera, to examine and analyze the 3D structure of the model protein. Pay attention to key structural features and interaction. Functional annotation. Consider adding functional annotation to the model if information about active sites, ligand binding site, or functional domain is available. Further analysis. Depending on your research goal, you may conduct additional analysis such as molecular dynamics simulation or docking studies to explore the dynamics and interaction of the model protein. Publication and communication. If your homology model is used for research or publication, provide details about the modeling process, including the template used, alignment method, and validation results. Homology modeling is a valuable tool for predicting protein structure when experimental data are lacking. It is widely used in structural biology, drug discovery, and understanding protein function. However, the accuracy of the model heavily depends on the quality of the templates and the accuracy of the sequence alignment. Structural modeling process often involve human intervention because the human expert knowledge combines with biochemical information, that is functions, mutagenesis, catalytic residues, etc., could help in the structural assembly and model selection. Development of fully automated algorithm allows non-expert to generate a structural model for their own sequence through internet services. Prediction of three-dimensional protein structure from amino acid sequence represents one of the most important problems in computational structural biology. ITASSER as a Zhang server was ranked the #1 server in recent critical assessment of structure prediction CASP and CASP 8 experiment. ITASSER is a widely used computational protein structure prediction tool that is used to predict the three-dimensional structure of protein from their amino acid sequence. It combines techniques such as homology modeling, ab initio modeling and structural assembly to generate a structural model of protein. Here are some key features and aspects of ITASSER. First one is the protein structure prediction. ITASSER is primarily used for predicting 3D structure of protein. Given an amino acid sequence as input, it generates a 3D structural model, providing valuable insight into the protein's potential folding and functional characteristics. Template based modeling. ITASSER uses template based modeling approach where it identifies known protein structure in the protein data bank that are structurally similar to the query protein sequence. It then uses this template to build a preliminary model. Ab initio modeling. If there are no suitable templates available for a given protein or a particular region of the protein, ITASSER employs ab initio modeling techniques to predict the protein's structure from scratch. This is especially useful for novel or unique protein folds. Iterative refinement. The iterative part of the ITASSER refers to the refinement process. It iteratively optimizes the structural model by using simulated annealing, molecular dynamics, simulation and other techniques to minimize energy and improve the model's accuracy. Structure assembly. ITASSER assembles a structural fragment obtained from the threading and ab initio modeling into a final 3D structure. It considers the spatial arrangement and interaction between these fragments to generate a coherent and biological relevant model. Functional annotation. In addition to predicting the 3D structure, ITASSER provides functional annotation and predicts potential ligand binding site, active site and functional residues within the protein. Web server. ITASSER is available through a user-friendly web server, making it accessible to researchers without the need for extensive computational resources or expertise in structural biology. Validation and scoring. ITASSER provides various scoring functions and quality assessment measurement to help user evaluate the reliability of the predicted protein structure. These metrics helps user assess the accuracy of the model. Validation and analysis. ITASSER provides tools for visualizing and analyzing the predicted protein structure. User can examine, model and download them for further analysis. CASP participation. ITASSER has participated in the critical assessment of the structure prediction competition, which is a biennial competition where different protein structure prediction methods are evaluated against experimentally determined structure. ITASSER has performed well in CASP assessment, highlighting its accuracy and reliability. ITASSER has been widely used in structural biology, bioinformatics and drug discovery research. It can be a valuable tool for researchers seeking to understand the structure and function of protein, especially when experimental methods like X-ray crystallography or NMR spectroscopy are not feasible. It can be particularly valuable for studying protein with unknown structure or for refining models of protein with low sequence identity to known template. However, like all computational methods, the accuracy of the prediction may vary depending on the complexity of the protein and the availability of the suitable templates. Researchers often use multiple prediction methods and experimental data to validate and refine the predicted protein structure further. This tutorial will give you to predict tertiary structure of protein by threading using ITASSER web server. Online service system in ITASSER contains online server for folding, docking design, domain etc. Some are downloadable. Bioinformatic tools like alignment, image, clustering etc. all are downloadable. Databases like ligand, GPCR, genome, decoy, potential, CASP etc. all are downloadable. Questions and issues can be reported and discussed in the services system discussion board. Resource required for ITASSER. Hardware, a personal computer with internet access. In case of software, only a web browser. To facilitate the management of modeling data and resource assignment, users are required to register their institutional email address at http://zanglab.ccmb.med.umich.edu/itasser registration.html. After the registration, a password is sent to the user which allows the user to submit and manage his or her files. The minimum input to the server is the amino acid sequence of a query protein in FASTA format. ITASSER is a computational method and server used for protein structure prediction. It combines various techniques to generate accurate 3D structure models of protein based on their amino acid sequence. Here is an overview of ITASSER method. First one is threading or fold recognition. ITASSER begins by identifying potential template structure from the protein data bank or other structural database that share sequence similarity with the target protein. This is known as threading or fold recognition. These template structures serve as starting point for modeling the target protein. Structure assembly. The method assembles the initial 3D structural model by aligning the target sequence with the selected template structures. This alignment helps in determining the initial backbone structure. Fragment structure prediction. ITASSER predicts the local structural features and conformation of protein segment using fragment based modeling. It breaks the target sequence into smaller segments and predicts the 3D structure of these fragments. Iterative refinement. One of the key strengths of ITASSER is its iterative refinement process. It generates multiple structural models of the target protein, often referred to as the decoys. These decoys are subjected to energy minimization and structural refinement in multiple iterations. The refinement process aims to improve the accuracy of the model by reducing steric clashes and optimizing side chain conformation. Scoring function. ITASSER employs multiple scoring functions to assess the quality of the generated model. These scoring functions take into account factors such as energy, stereochemistry and structural consistency. The model with higher scores are considered more reliable. Model selection. Based on the scoring and refinement, ITASSER select the best structural model from the pool of generated model as the final prediction. The confidence in these models is often indicated by the C score, where higher values represent higher confidence. Functional annotation. ITASSER can provide functional annotation for the predicted protein structure. These includes information about potential ligand binding site, active site and gene ontology terms that describe the protein's molecular function, biological process and cellular component. Output. User can assess the result including the predicted 3D structure in PDB format. Via the ITASSER web server, additional data and analysis are often provided, allowing researchers to gain insight into the structure and function of the target protein. ITASSER is a valuable tool for structural biologists, bioinformaticians and researchers interested in protein structure prediction. It is especially useful for predicting the structure of protein with no closely related experimental structure available and has been widely used in various areas of molecular biology, structural biology and drug discovery. Analysis of the result. Analyzing the result obtained from ITASSER, a protein structure prediction tool, is a crucial step in understanding the predicted protein structure and its potential biological significance. Here are some steps and consideration for analyzing ITASSER results. Start by retrieving the result from ITASSER server, which typically includes a set of predicted 3D structural model for your target protein. These models are usually ranked based on their estimated quality score. Select the base model. Review the quality assessment score associated with each model. ITASSER often provides scores like C score, that is confidence score, TM score, structural similarity score. Select the model with the highest C score or the best TM score as the initial candidate for your analysis. However, it is initial to assess the consistency of these scores across multiple models. Visualization the structures. Visualize the structure. Visualize the selected model using molecular visualization software such as PyMol, UCSF Chimera or JMol. This allows you to examine the 3D structure in detail. Pay attention to the overall fold secondary structure and potential ligand binding site. Evaluate model quality. Assess the quality of selected model using various structural validation tools. Some commonly used tools include Ramachandran plot. Check the distribution of the dihedral angles to assess the stereochemical quality of the model. Mol probability. Analyze classes, bond angles and bond length to ensure geometric accuracy. Verify 3D. Evaluate the compatibility of the model with the amino acid sequence. PROSA or other energy based scoring method assess the energy and quality of the model. Compare with templates. Compare the predicted model with the template structure used by ITASSER for modeling. Identify region of the structure where the model closely matches the template and regions where it differs. Functional insight. Analyze the predicted structure for potential functional insight. Look for binding site, active site and regions that might be involved in protein-protein interaction. Consider the implication of the structure in the context of your research goal. Structural alignment. Perform structural alignment with known experimental structure if available to assess the accuracy of the prediction and identify structural similarities or differences. Ensemble models. ITASSER often provides an ensemble of models. Consider analyzing and comparing multiple models to capture structural variability and uncertainties. Publication grade figure. If you plan to use the predicted structure in publication or presentation, create high quality figure that effectively convey the structural information. Interpret the result in context. Consider the limitation of protein structure prediction. It is essential to understand that ITASSER provides a model based on computational method and the accuracy may vary depending on the target predictions characteristics. Further experimental validation. Depending on the significance of your findings and the intended use of the predicted structure, you may want to perform experimental validation such as X-ray crystallography or NMR spectroscopy to confirm the structure. How to submit a query sequence in the ITASSER web server. Use of ITASSER is very simple. Just open ITASSER server and it simply need to copy and paste your query protein sequence here in this box, and then you have to provide some of your credentials such as your academic email ID, password. To get the password to submit your job in ITASSER, you need to first register yourself with your academic email ID in ITASSER server and ITASSER will send you the password. You can use this password when you are submitting this job. After giving your credential and your sequence, simply click on this run button in ITASSER. After some times you will have 3-dimensional structure. When the job will be done, an email will be sent to you. In this email there will be a link. When you click on this link, you will be directly land to this page. Understanding of this result is very important, in which form the ITASSER has sent you the result. The first thing which you will note here in this result page is your query protein sequence, which you have submitted to the ITASSER server for the prediction of 3-dimensional structure. Second thing is the information about the secondary structure of your query protein. Here you are going to have an idea about which part of your protein is going to make alpha helix and which part of your protein is going to make beta sheet and which part is going to make coil. Secondary structure is represented in alphabet. Below the sequence, the H represent the helix, capital C represent the coil and S represent the sheet. Below this alphabet, you can find out some confidence score. If the score is near to 10, the prediction accuracy is very high, and if the score is near 1, ITASSER is not sure that particular polypeptide parties make this type of secondary structure. Then in next section, you are going to have an idea about solvent accessibility. Solvent accessibility let you know that which amino acid is present at the surface of the protein and which amino acid is present at the core of the protein. If the solvent accessibility score is near to 9, it means that amino acid is present at the surface of the protein. If the score is near to 0, means these amino acids are present in the core of the protein. As you know that amino acids which are hydrophobic in nature, they are normally buried inside the protein, while the amino acids which are hydrophilic in nature, are hydrophilic or polar in nature, are present at the surface of the protein. Then you are going to have an idea about B factor. It tells about the flexibility of your protein. Flexibility means how much kinetic energy of your amino acids are going to have. In this graph, if the scores are below 0, it means that these amino acids are very rigid. They have less kinetic energy. If the scores are above 0, the amino acids are flexible. In our protein, first few amino acids have high positive value. It means that these amino acids are very flexible. After the B factor, you are going to have an idea about the template which you are used for making the 3-dimensional structure of your query protein. Here in these template, the normalized G score important value. If G score is above 1, then this template has good alignment with your query sequence. Keep this value in view. If the score is less than 1, then the alignment is poor. If this template is used in making 3-dimensional structure of your query protein sequence, then this structure could be suspicious. After the template sections, you are going to have an idea about your 3-dimensional structure of your query protein sequence. ITASSER will predict normally 5 different models. Sometimes it produces 3 models. Among these models, you have to select some appropriate model. Normally the model are selected on the basis of C score. Higher the C score is better will be your model. But the C score is not only the criteria on which you select your protein structure model. There are many other things as well which we will talk about later, on which you should select your protein structure. Below the 3-dimensional structure predicted model, we are going to have an idea about alignment of our predicted structure with the template. Here you can see these are the templates which are used for the making prediction about 3-dimensional structure of your query sequence. Let's assume that if I want to align 3D structure predicted by ITASSER with the template, I can simply click on this radio button. I will have an idea that how much my predicted structure is aligned with the template, means how much it is similar with the template. You could have good idea about the structural similarity of predicted structure with template structure. Below the alignment section, you are going to have an idea about ligand binding site which could be present in your predicted structure. Here in this 3D structure, you can see some pink things which are ligand binding site residues in your query protein. In the next section, you will be having an idea about the enzyme and active site which could be present in our protein. Our protein is not an enzyme, that is why we are not having any idea about the active site. But if your protein is an enzyme, you could also have an idea about what are the residues which are making active site in your protein structure. So this is all about ITASSER result. Hope this information will be beneficial for you. Thank you.